And I'm asking this because I'm teaching. I teach yesterday also. Okay, you can still do those experiments. Agree, but there is more direct method. Which nucleosome modification? He's told, right? One modification is S3K9 trimethylation. So how will you answer this question? So you have to remember that what are the modification which corresponds to heterochromatin. So yesterday I teach you DNA methylation is also important for heterochromatin. S3K27 trimethylation is also important for heterochromatin. So you can probe those two also. That was the answer I was looking for you. Did I not teach yesterday? DNA methylation I was teaching yesterday, heterochromatin, chromosome inactivation. So whatever I will ask, I will not ask out of box. I'll ask, if you've gone through my lecture very carefully and mindful way and thought process way, I'll ask from there on. So this group actually used S3K9 trimethylation as the technique, and they used very fancy things which is called stochastic optical reconstruction microscopy. So I put a layout here, but since this is the main aim of this spectroscopy is you can get the better resolution. If you do the simple microscopy, the resolution will be not better, but if you go to this microscopy, resolution will be better. So you can study that. So what you can do, like when you do the storm microscopy, you can see that in wild type you get a lot of signal of S3K9 trimethylation cluster, and as tumor is progressing, your signal is gradually spread out. Intensity is also reducing, like see in the accelerated here. These are the zoom images of this. So if you focus on this, this is very high intense, this is little bit less, this is little bit less, but this one where tumor is happening very accelerated way, you see the less intensity if you compare with this, and that has been quantified over here, so you can quantify through this. Yes.
Yesterday you have told that heterochromatin is present near the periphery. So how it is different from the middle one where we are seeing the tumor? Like if tumor is present, that is like because of the peripheral heterochromatin also. So how can we differentiate in between heterochromatin and peripheral?
So you are confusing with two things. One is this images is just we are probing in the whole cell. So you can see here also, here also, here also. These are the periphery. So people have quantified this one also. It is not here. So mostly in periphery region you are seeing the signal, right? This one, this one. The box has been drawn to amplify this, but you can see in the periphery it is there, right? The bright is means more S3K9 trimethylation is there. So it is in periphery, right? Yes. Yeah. In periphery is also there, right? Which is very less here. This paper has a lot of other. So this is one way to show. So clearly you got the experiment. This is for the same thing with the another cell type. This is for this is taken from the human samples, human patient samples, and the previous was from the mouse sample. So here also you see the same thing. These clusters are of reduced size in advanced adenoma where the cancer is high. So this is where this suggests that you can heterochromatin is involved in the cancer phenotype. So this one is from human sample, the previous one was from the mouse sample. Now we come to this chaotic slide which you know. So now you understand that heterochromatin is involved. So if you can inhibit heterochromatin formation, if you design the drug, then you can basically make the cure against that. So people are saying that actually chromatin state, which consists of all this activity, is important for the particular transcriptional status of the cell. So two things happens back in there. People will come with the histone code hypothesis, which you might aware of, and also people say that one function correlate with the another. So for example, if I say that H3K27 trimethylation, or this one, this is 9, is important for heterochromatin formation. So wherever you have K9 trimethylation, it will be heterochromatin. Over K27 trimethylation, it will be heterochromatin. And there is another mark here, is called, if it is phosphorylated at this position, there will be no heterochromatin. And sometime these modifications work in a concerted way. Okay. So you are here that, sir, now we get totally confused. The confused state is, I said that K27 trimethylation is for heterochromatin, but somewhere combinatorial modification also exists to give the phenotype. So many times a modification does the two different function in the cell. For example, H3K36 trimethylation. So H3K36 trimethylation is important for transcription, elongation, but H3K36 trimethylation is important for DNA damage response also. So different modification could do the different thing. And if you try to read all the modification type, you will be totally fed up. मुझे हेट्रोक्रोमैटिन, न्यूक्रोमैटिन, एपिजेनेटिक्स नहीं करना है सर, मुझे फेंक दो सब, दिमाग खराब हो गया मेरा, मुझे कुछ नहीं करना है. Because the field is so emerging that sometime methylation is corresponds with activation mark, sometime methylation is for repression, sometime sir, you are saying that this also exists, this also exists, it's make the chromatin states, and in fact there are 9 or 10 chromatin states has been reported in cancer cells. So the field is very much things, but as a student you want to go in the field, right? Then you have to go slowly. So I will say that first focus on these 4, 5 residue: K4, K9, K27, K36, K79, K20. If you just think about those, take any one of this and learn the whole process. If it is required in your career, then you can go for novel modification, because these things has been very well studied. Although it has been well studied, but still I will tell you that why we do not have still the good therapy. At the end of the lecture, some point of time I will tell you. Okay, so sidewise try to make the notes what I am saying, because if you just live on the slide, then you might miss many things. Many things I am telling you, so those who can make a good note will be benefited later on, because these are all taken from the review papers, from books. So it's not going to help you. All my slides, even if I gave you, it is not going to help you out. But the additional things I am talking in between, that which is actually helpful. And that's why I am here, because you have the artificial intelligence, chat GPT, different things. What is my need then? So my need is to understand. So why I said that, if I go to the back slide, see, I just put a few examples. Let me go to the back slide, then you will understand. I'm just talking about H3. You have a H4 also, you have a H2B also, you have H2A also, and then you have a H3 variant, H2A variant, you have a different chaperones. Think about how complexity is there. And that complexity is essential, because I told you in the first slide, I showed, right, you have a zygote. And from zygote, you are getting neuronal cells, you are getting liver cells. Remember my first slide for the today's thing. You have to get different cell type from the same thing. So this complex thing is only possible if there is a complexity at the epigenome level. That's point I'm trying to give you. And to go into this complexity, you need some toolkit, to need some your thought process, and that is why I'm here, to give you that thought process. So the thought process is, sir, you are talking about so much modification. So that's why I said that don't bother about all this. When required, I will read it. But bother about the one which I showed you. Why? Because this has been most studied and more correlative. Clear? So that is my reason I come for K4, K9. Now K4 can go acetylation as well as methylation both. So yesterday I have that question. I told that today I will going to talk about that. So yesterday I have that question. Use higher eukaryotes versus low eukaryotes. I asked that what are the model system where epigenetic mechanism have been studied. And I said that you can go to yeast to Drosophila to human, human to Drosophila to yeast. And there is advantage, disadvantage to this system. I asked that question, remember? Those who were here. So this slide actually explains that conundrum. What it's telling that this color is yeast. So in yeast, for example, we also study Saccharomyces pombe in our lab, apart from the so-called cell line, mouse, cancer phenotype. All those are very attractive to different people when they apply immunotherapy. Whenever I talk to students, sir, हमें तो immunotherapy करना है, हमें तो cancer करना है. Whenever I talk about yeast, they have like little bit down thing. But I'll tell you what are the advantages and disadvantages. Because sometimes this system is better, sometimes other system. Of course, if I am probing the cancer phenotype, I will not say that go for yeast, right? So it depends on what context and what questions you are asking for your PhD or MSc thesis. So what is here, like, let me focus on one because I'm talking too much about K9 and K27. So I will focus my discussion on this only. If I'm able to show this and these two, then my job is done. If you understand how to design an inhibitor against this. So in yeast, you have only one enzyme. This is called CLAR4. This enzyme does K9 trimethylation. But if you see in the Drosophila, you have a different variant, G9A, ZDB1. All these enzymes, all these genes has been shown to have a K9 methylation activity. And in human, सर यहाँ तो और भी दिमाग खराब हो रखा है. यहाँ सुबह 3-9 आप लिख लिया है, H2 भी लिखा है, G9A भी लिखा है, GLP भी लिखा है, ZDB1, PRDM1, जिसपे थोड़ा सा क्वेश्चन मार्क है, कुछ लोगों ने दिखा है, कुछ ये तो और भी कॉम्प्लेक्स सिचुएशन है. मैं करूँ तो क्या करूँ, right? You excited that okay, I found it, K9 trimethylation is there in your gene, and you say, sir, it looks like heterochromatin. You did the chip experiment, chipstick experiment, Western blotting, send the paper, and the reviewer said that no, we want to know which methyltransferase is important. And then we choose in the human, you have so many methyltransferase. Sir, you showed that K9 trimethylation in the previous slide is important for tumor. Now I'm going to cure the tumor because K9 I found it. So now you go for the designing experiment, you have so many enzymes, does the same thing. So somebody say that, right? I'll design the inhibitor, sir, it will going to affect other reader eraser. Of course it's going to affect because the same enzyme has doing the same thing. So the biggest problem in all this thing is to find out the specificity, because I'm just talking about K9 trimethylation. K9 trimethylation होने से पहले you need to have a K9 dimethylation, K9 monomethylation, फिर K9 itself. 4 चीजें आ गई, मैंने बोल दिया फटाफट. K9 itself, K9 monomethylation, mono means only one methyl group is there, K9 di and tri. So I come to the morning lecture. Sir, prove it by in vitro experiment. K9 trimethylation is important, not K9 dimethylation. So if you are able to purify histones from the cell line, are you going to answer? Because the cells, if you are doing from the human cell line, it has all the enzymes. So one way is to do the CRISPR, do SUBA3 and I CRISPR, and then see by Western blotting if you change in K9 trimethylation or not. All us know we can do the CRISPR of this. You can also generate antibody against dimethylation. You can do that also to probe the specificity. So sometime, I'm just talking about this practical situation. You have make a knockout of SUBA3 and NH1, and you have make a knockout of GLP and see that, for example, I'm telling, I don't remember exactly what is what, but the point here I'm trying to make that if you knocking out your enzyme and if you do the Western blot, still you see that level is very much similar in the knockout and wild type. This is telling us that there could be redundant enzymes which is doing the same function. Again, this can be interpreted in another way also, because overall K9 trimethylation is not changing, but it could possible that certain genes, you understand my point, right? It could be a localized effect. So if you do not see the overall change in K9 trimethylation, you do the ChIP-seq. So how many genes where you see the loss of K9 tri and dimethylation experimentally? In this way, you will go your experimental approach. Okay. So it's clear or not clear? And then you can test your inhibitor. So there you can answer all these questions, whether you can do double knockout, triple knockout. Many times they will not survive. The advantage in yeast was, I'm coming back, since there is only one enzyme and this was not essential. So people have knockout CLAR4. If you knock out CLAR4, then you make a strain without this enzyme, and if you do to test by Western blot. In the cell उसमें कुछ दिखा ही नहीं. And that marks in storm I showed. The tissue is very indistinct. Staining show nothing is going on. But figure it out that there is a chip by using ChIP-seq working out. अभी हमारा is clear. I am not going. Okay. Now, but he is the one who is father figure in the area. CD Alice, BD Stoll, these are all the big people who have worked in the epigenetics, and the thing has been initially discovered in East. Whatever you are extrapolating today in the cell line, remember those had been done either in East and Drosophila to figure it out. What is what is not there. So people come with this thing that, okay, what is there is there is no relevant of East system. There is no relevant of those, especially for the new generation, because now you have a fancy technique like CRISPR. You can easily do all these experiment in the cell line. Okay, but they all have been discovered in East in those days, because this thing similar to thing today we have the mobile phone, but on other day it was not there. Okay, but if you want to learn something, you have to go to the discovery, and then your concept will gradually develop, which is lacking in, I find, as a research institute.